Osmosis in raisins. In this module, you will learn how osmosis takes place in raisins. Osmosis is the movement of water molecules from a region of higher solvent concentration to a region of lower solvent concentration across a semi-permeable membrane. In our body, various nutrients are transported through osmosis. Since it is difficult to observe osmosis in our bodies, let us learn about this process by observing it in raisins. The materials that are required for this activity are a beaker, water, concentrated sugar solution, and raisins. Let us begin the activity. Take a beaker. Fill half of it with water and put in 5 or 6 raisins. Leave them undisturbed for about 4 to 5 hours and then observe any changes. What do you observe? Hey, all the raisins in the water have swollen. Their size and volume has increased. They look much bigger than they originally were. Now, to understand the process better, let us do another small activity. Take another beaker filled with concentrated sugar solution. Now add the swollen raisins in it and leave the beaker undisturbed for about 4 to 5 hours. Now, let us observe the raisins. Look, the raisins have shrunk. This means that the size and volume of the raisins has decreased. Now that you have observed the changes in both the cases, let us look at the explanation for these changes. In this activity, the raisin resembles a cell, and its outer membrane acts as a semi-permeable membrane which allows water molecules to move through it. Consider the first case where the raisins were kept in water. The water has a higher solvent concentration than the solvent concentration inside the raisins. Now, due to difference in concentration, an osmotic gradient is set up, which results in water molecules moving from the water into the raisins. This type of osmosis is also known as endosmosis. This is why the raisins, when kept in water, swell up. On the other hand, in the second case, where the swollen raisins were kept in concentrated sugar solution, they shrink. Here, the outside medium has a lower solvent concentration than the medium inside the raisins, which means that here sugar solution acts as a hypertonic solution. Due to the difference in concentration, exosmosis occurs, which causes the water molecules to move from the raisins into the sugar solution. It results in the raisins shrinking. From these activities, you can conclude that when raisins are kept in water, endosmosis occurs, due to which water enters the raisins and causes them to swell. However, when raisins are kept in concentrated sugar solution, exosmosis occurs, due to which the raisins shrink and become flaccid. In this module, you have learnt that osmosis is the movement of water molecules from a region of higher solvent concentration to a region of lower solvent concentration across a semi-permeable membrane. When raisins are kept in water, they increase in size due to endosmosis. When raisins are kept in concentrated sugar solution, they shrink due to exosmosis.
Study of osmosis. Osmosis is the process by which fluids move from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. Osmosis occurs due to the free energy of the solvent molecule in two regions. The free energy of the solvent or pure water is greater as compared to that present in the solution. So when osmosis occurs, the solvent moves from a region of its high free energy to a region of its low free energy through a semi-permeable membrane. To do the experiment, we require a large potato without peel, knife, 20% sugar solution, Petri dish, distilled water, pins, and a tile. Procedure: take a potato from the watch glass and place it on the tile. Take a knife and cut both ends of the potato to make it flat. Using the knife, make a cavity in the center of the potato from the flat side almost up to the bottom. Take a beaker containing distilled water and pour the Petri dish half full of distilled water. Now, place the potato on the Petri dish that is half full of water. Take the beaker containing sugar solution and fill half of the cavity of the potato with 20% sugar solution. Mark the level of sugar solution in the cavity with the help of a pin. The potato now functions as an osmometer. Leave the osmometer undisturbed for about 2 hours. Mark the rise in the level of sugar solution in the cavity with another pin. Observation: the level of sugar solution in the potato cavity rises after some time due to the entry of water into the sugar solution through selectively permeable membrane of the cells of the potato. Conclusion: the movement of water from the Petri dish to the potato cavity occurs because of the difference in the concentration of solvent molecules in the two regions, sugar solution in the potato cavity and pure water in the Petri dish. Precautions: the cavity should be deep enough to keep only a thin layer of tissues at the base of the potato. The sugar solution should be sufficiently high osmotic concentration as compared to the cell sap of potato cells.
Osmosis is the passage of water from a region of high water concentration through a semi-permeable membrane to a region of low water concentration. It is a special case of diffusion through a selectively permeable membrane. Let us see this phenomenon in action with this activity. Here we have 2 eggs and 3 beakers filled with dilute hydrochloric acid, concentrated salt water and water respectively. Deshell the eggs by dissolving them in dilute hydrochloric acid and wait for few days. Now, immerse one deshelled egg in water and the other in concentrated salt solution. Leave the beakers undisturbed for some time and observe. we see that the egg in the water swells as the water passes into it by osmosis. This happens because the egg has a lower concentration of water than the water in the beaker. On the other hand, the egg in the salt solution shrinks. Here, the water passes out of the egg solution into the salt water because the salt water has a lower water concentration. Hence, in this activity, we have seen two cases of osmosis with an egg, wherein water passes from a region of high water concentration through a semi-permeable membrane to a region of low water concentration.
Prepare semi-permeable membrane by using egg membrane. To perform this activity, following materials are required: 3 beakers, 2 egg membranes, sugar, water, thread, measuring jar, disposable syringe, cloth and tablespoon. Take the eggs that are soaked in the dilute HCl solution. Wash the eggs under tap water and wipe them with cloth. Now, remove the internal content present in the egg membranes carefully by making a pencil-sized hole in them. And wash the membrane with fresh water. Now take 1 egg membrane and fill it with 10 ml of saturated sugar solution, which is already prepared in a beaker by using syringe. Next, tie the mouth of the membrane with the help of a thread. Now, take a beaker containing 100 ml of tap water and dip the membrane into it. Leave it aside for 1 night. Next, take the second membrane and fill it with 10 ml of tap water with the help of syringe and tie its mouth with the help of thread. Take another beaker containing 100 ml of saturated sugar solution. Use the sugar solution which is already prepared and place the second membrane inside this beaker. Measure the contents of the egg membranes and beakers and note down the observations. It is observed that in case of beaker 1, the water level in beaker is decreased and the sugar solution present inside the egg membrane is increased. In case of beaker 2, the sugar solution present inside the beaker is increased and the tap water present inside the egg membrane is decreased. From this experiment, we learned that the level of solutions of egg membrane or beaker is increasing only when they have the sugar solution in them. The sugar molecules are too large to pass through the membrane. Hence, the water molecules pass into the sugar solution. This decreases the level of water and increases the level of sugar solution. This increase of sugar solution is due to osmosis across the semi-permeable membrane. Semi-permeable membrane allows only certain materials to pass through the cells.
Good morning, everyone. If someone is cooking something delicious, you would get its smell from few meters away, which means that smell spreads through the air. Now, we burn this kind of coils to repel mosquitoes, right? At first, You don't get the smell of burning coal unless you stand very close to it. But gradually the smell spreads throughout the room. After few minutes, you will get its scent from anywhere in the room. Means the smoke spread in all the direction. Same way, if you add 2-3 drops of ink in a glass of water, you would see how the ink spreads throughout the water equally after some time. What you see here is called diffusion. In diffusion, molecules of material spread equally throughout a medium like air or water from higher concentration to lower concentration. Because of this diffusion, smell, gas, smoke, etc. spread through air easily. Because of this process, if you add color in water, it will spread throughout equally. Because of diffusion, flavor of tea spreads throughout the whole water in a cup when you put a tea bag into the cup filled with hot water. But you know, diffusion is much more important than that. Oxygen-rich air reaches alveoli in lungs. When deoxygenated blood flows through capillary around these air bags, then oxygen diffuses into blood. This is because the concentration of oxygen is less compared to oxygen in the air. Many essential ions, atoms, molecules, nutrients, etc. spread from higher concentration area to lower concentration area in diffusion process. Hence, those get utilized properly. There are many other examples of diffusion inside a body. And don't forget, the process osmosis is also an example of diffusion of liquids. So you see,